Welcome to Let's Meet the Virologist, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with students, postdocs, and other virologists so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. On June 4th, 2022, we talked with Aaron Embry, a graduate student at University of Texas Southwest Medical Center in Dallas. He obtained his bachelor's degree in biology at Millersville University in Pennsylvania. He now studies host determinants of arbovirus restriction. Thanks for talking with us today. Why don't you tell us a little bit about yourself?
Yeah, sure. So first of all, well, thank you for having me. I'm actually a big fan of the podcast, and so I'm excited to be a part of it. But my name is Aaron Embry, and I'm from a small town, Spring Grove in southern Pennsylvania. And I went to undergrad actually at Millersville University in Lancaster, Pennsylvania. So it's only about 40 minutes or so from where I grew up. I didn't kind of branch out a whole awful lot there. And I got my bachelor's in biology with a minor in chemistry. And after graduating, then I worked briefly in industry for about 2 years. I worked 2 years while I was a junior and senior to help pay my way through college as well. So 4 years total. And then I decided I was going to test the waters with academia. And I worked there for 2 additional years before deciding that a PhD was what I actually really wanted to do. And so that puts me where I am today, a PhD candidate at UT Southwestern here in Dallas, Texas.
Great. And can you tell us sort of how you chose your particular PhD institution and then how you chose your lab? People are often interested in that. What are the choices? What attracted you to those places?
Oh, man. Yeah, it's been quite like the long, strange trip, really. So, well, after I graduated undergrad, and I briefly mentioned there that I worked in industry for 2 years, it was pitched to me to potentially move to DFW or the Dallas-Fort Worth, Texas area. And I decided at the time, this was 4 or 5 some odd years ago, that I was young, dumb, and broke enough to make that move and pick up and explore what else was out there and have some adventures outside of just small town Pennsylvania. And so I moved to Dallas, and in doing so, I needed to find a job, and so I needed to pay rent, eat food, and I actually posted on Reddit what the big hiring agencies were in the area, but what big science or tech places were around. And essentially, the 2 major answers I got were Alcon, which is industry, or UT Southwestern, which was academia. And so given that I was really interested in potentially pursuing academia more, I decided I was going to apply mostly there. I did dabble a little bit in maybe going to industry. You're trying to make sure you can cover your bases and have backups. But I was really lucky and really fortunate. I got hired in the immunology department under Dr. Robert Orchard. And I told Rob at the time that I was interested in potentially pursuing a grad school further later. And he said, okay, if that's what you think you want to do, then I'm going to treat you like a student. I'm going to give you your own project. I'm going to make you present at lab meetings or floor meetings with the other departments. You're going to make posters, go to talks and symposiums and stuff. And so I did all of that. And I really had an absolutely amazing time. It really solidified to me that a PhD is what I wanted to do. And so after the 2 years that I told Rob I would help establish the lab, getting projects going, recruiting some students and stuff. So Brittany Stewart was actually hosted on this podcast back in episode, I think 35 or so. After helping everyone get established, I then applied to grad school myself, and I applied here at UT Southwestern as well as a couple other places back in Pennsylvania. So that's where my family still was, and I had considered maybe wanting to move back, but I got in here at UT Southwestern and decided that I was interested in staying. I didn't want to go back just yet, and I wanted to explore what other people on campus were potentially working on rather than just staying in Rob's lab. If I didn't like what other people were doing, maybe I could circle back around and ask Rob about rejoining. But he kind of pushed me and said, go out there and see what else is there. You really only know me so far. And so I did. I reached out to actually an old collaborator, some projects that I worked on when I was in Rob's lab, Dr. Don Gammon, as well as Rob's old PI and mentor, Dr. Neil Alto. And I reached out to them individually, but they actually kind of met outside and then came back to me together and pitched that I potentially do this joint mentorship between the two of them because they had recently received some funding for this really interesting project that kind of spans both host innate immunity as well as bacterial infections and co-infection with viruses and stuff. And so I rotated in both of their labs here at UT Southwestern. Your umbrella program, you don't really have to pick right away. You can kind of test the waters with a bunch of different stuff. And so I tested the waters with bacteria, because I hadn't really done that. I've been mostly a virologist up to this point. And I actually really enjoyed it. I fell in love. I was kind of hooked at all the moving parts there. And so I joined, and that's where I am now.
Cool. And I guess I forgot to ask you, way back when, have you always been interested in science, or was this something that just kind of developed gradually?
Yeah, that's a pretty great question. As far as I can remember, I've always had a knack for it. My mom and my grandma like to share this picture of me dressed up in like a white coat as a doctor when I was like 2. And so, I mean, growing up, unfortunately, I'm relatively dyslexic. So I've already kind of struggled with reading and writing. And I feel like when you're young, you tend to gravitate towards the things that you're good at. And for me, that was mostly math and science. And so despite the fact that I was good at those, I'd still kind of coasted through high school without really a whim or an idea of what I wanted to do next. Until I had in my senior year, I had this chemistry class with this really incredible teacher, Miss Kimber. And she just was so patient and colorful and really helped foster my interest in science, and all science for that matter. And it was around that time that I decided, I was going to go to undergrad for bio and chem. But when I went to Millersville, generally situated in the hills of Pennsylvania, is not a virology hub. It's mostly a lot of agricultural science. So unfortunately, I didn't have a lot of opportunities to get involved in virology there until actually I came to Texas and was in Rob's lab. And that's where I kind of developed my niche and experience with viruses, and mostly influenza in his lab, but a little bit of the MNV work, which is a mouse norovirus, murine norovirus. And that was where I kind of started the virology track. And now it's just kind of taken me by storm.
Yeah. And can you tell us a little bit about what it's like having co-mentors? So that's a little bit more unusual in science, although I think it's getting more common now. So tell us what that's like.
Yeah. So my current lab, the Gammon lab, has me as well as 3 independent students that are just to him. And then in Neil's lab, he's got 2 independent students now. We just got another one. And then 2 co-mentored students as well. And he's dabbled a bit more in this co-PI ship as our university likes to tote during recruitment and other things. We are a really collaborative work environment, and a lot of the PIs talk to each other. It's a really wonderful place to be. And I really like having 2 PIs. It has its struggles because it's double the people to potentially mess up communication with or miss a meeting with that you didn't realize. But it's also double the people that are really excited when your project is going in a really cool new direction. And it's double the people to bounce ideas off of. Neil's lab in particular is a little bit more developed, and so he has more postdocs. So that's some older, more established scientists I can go to with more technical problems. But Don's lab has more grad students that I can connect with on like a personal level and just like be friends with and go out with. So that's really, really interesting. But everyone also knows that I'm a relatively social person here. So I kind of dabble around a bunch of PIs. I like to say that I collect them like Pokemon cards. So I go around and talk to all the people on the floor and interact with everyone really. So having 2 PIs doesn't really necessarily feel all that different than 1 PI to me. It's mostly just double the meetings is the major negative.
Right. So I guess then why don't you tell us a little bit about your thesis work, sort of like the bigger picture, and then maybe some of the types of experiments you're doing, the techniques that you're using to answer those questions.
Sure. Yeah, that's the fun part, right? The virology and what we're excited to talk about when we go to ASV this year. And so boiled down, like I kind of mentioned earlier, my project has 3 moving parts, right? We have the host, the bacteria, and the virus mostly. And so kind of starting with the virus-host interaction, I primarily study arboviruses, which I feel like for the case of ASV, most people know what that is, but in case anyone doesn't, arboviruses are an informal classification of viruses that are essentially anything transmitted to a host through an arthropod vector, right? So that includes fleas, ticks, biting flies, and then of course your friendly neighborhood mosquito. And for me, I primarily study VSV, Ross River virus, Young virus, and Synvis virus. So that's 3 toga viruses and then the rhabdovirus. And despite these viruses being really, really adapted to infecting dipteran insects, so that includes the flies and mosquitoes that I mentioned earlier, they do not actually establish very productive infections in lepidopteran cells. So that's mostly moths and butterfly cells. And the curiosity there is the fact that these 2 orders of insects are actually really closely related. And so the barriers between them two, when you think of the case of like innate immunity, hasn't really been investigated. And so the Gammon lab in particular is really interested in uncovering the differences in those 2 systems and what barriers to viral infections are there in a lepidopteran cell. And so previously, actually, he showed that if you co-infect these restricted arboviruses with a large DNA pox virus like vaccinia, so vaccinia virus, relatively well-studied pox virus at this point, you can actually rescue the restricted arbovirus replication under a co-infection system. And that's going to be this actually the talk by a fellow student, Dahi So. She's also presenting at ASV this year. And so if you're interested in that, go check her talk out. But my project is really taking that to the next step and saying, can we rescue these viruses now with bacteria? And that's where my other mentor comes in, Dr. Neil Alto. And his lab at its core is really interested in trying to understand the dynamics of these proteins that are used by bacteria during infections, known as bacterial effector proteins. And these effector proteins are secreted into a host cell through usually a needle-like apparatus, such as a type 3 secretion system, either before or after in infection. And these effectors have been shown to modulate a myriad of host cell interactions. You got immune evasion, membrane ruffling to promote uptake, all kinds of stuff. And so the majority of the bacteria that I use in my study actually study intracellularly. So that's the same place, if you think about it, that the virus itself is trying to replicate, right? And so we hypothesize that they're likely encountering a lot of the same or similar barriers, and obviously some other ones because you're 2 different pathogens, but they're all trying to really modulate the same host cell environment. And so we were interested in potentially looking at what those bacterial effector proteins could be doing that might be able to rescue my virus. So if you think about it, a bacteria has way more genetic real estate than my small 11 kb VSV genome. It can encode for so many more things. And so what we did was we cloned out a plasmid library of over 200 bacterial effector proteins, most of which with currently relatively unknown functions, don't really know what they do yet. And it hopes to screen them then against these restricted arboviruses for any effector protein that might be able to rescue that infection. And what I identified was a myriad, roughly, I think 20 or so for each individual virus. So the 4 that I mentioned earlier. But what really stuck out was these roughly 6, what I call core effectors, which were able to rescue all 4 of the viruses that I screened. And so they obviously are targeting something that's really important to both a bacterial infection and a viral infection. And my work has really been honing in on one of those top hits, which is this IPA-H effector, which is encoded for by Shigella. And so I tend to use a lot of viral rescue assays, right? So the co-infections and seeing if I can get infection when I overexpress or delete something, as well as given the fact that this IPA-H effector is a known family member of bacterial E3 ligases, I tend to use a lot of ubiquitin-related experiments. So from note, it would be like ubaits, which are ubiquitin-activated interaction traps that you can use to kind of screen hosts lysates for potential substrates of your ligase. As well as degradation assays. So if you express the ligase with a potential target, does it get degraded? And then, of course, if you add back things like MG132, can you mess up that degradation? And then most notably, I think the gold standard right now for the field is like in vitro ubiquination assays. So if you purify both proteins and you mix them in a controlled environment, do they still get ubiquinated? Is that a direct target, or was that degradation and off-target effect of maybe what the real target is? So that's kind of mostly the things that I'm doing right now.
Great. And the bacterial proteins that you're talking about, is this more a tool to understand sort of the virus host interaction or the bacterial host interaction? Or are you actually thinking that sort of in nature, there might be this interaction between all three?
Yeah, I guess given the context of the viruses that we're studying, a real natural interaction between, let's say, an arbovirus and a gut pathogen seems relatively unlikely.
Right. It'd have to be, I guess, in the mosquito, right? So not so much in the human host, but maybe, I guess, in the mosquito, you could have interaction, maybe.
Well, yeah, I mean, that's kind of where I parse down that we do just have so many moving parts for this project. And it's honestly, I mean, the blunt way of answering that question is I tailor what I say is the tool based on what my audience is. So if I'm at a bacterial conference, I'm talking about, I'm interested in this IPH effector and how that can be used during bacterial infections, and look at this virus helping me tease out those interactions. And then here at ASV, I'm really interested in the viral interactions and look at this bacterial effector that was helping me interact and perturb host cell systems that we don't yet understand all of the intricacies behind. It was only very recently that a lepidopteran genome was even published in the literature. So we really were limited in what our resources were to perturb this system. And so these are like tools and mechanisms that we can use to kind of get around all of these blockades and really get to the nitty gritty biology underneath.
Right. And do you think that some of your core bacterial effectors could actually generate permissivity to viruses that don't even infect either of your your insect host species?
Potentially, yeah. Some of the things that we're looking at also are the fact that I noted VSV as one of my 4 viruses, and VSV G protein is a common protein for like lentiviral work and stuff. And a lot of this work right now in lepidopteran systems is kind of inhibited for lentiviruses because that VSV-G protein, which is usually coding lentiviruses, can't infect these cells. So the things like CRISPR and other larger screens are way more hindered and a lot more difficult to conduct. And so we're also kind of thinking that this is stuff that might be able to be used as more of a method to making those experiments a little bit more permissive in the future, which would really help open up a whole avenue of new interactions and areas of study potentially in the future if we could help establish those kinds of tools and protocols for people in the future.
Right, great. And so thinking, I guess, in the future a little bit more, now that you've been a PhD student for a little while, what are your thoughts sort of like in the short term and then in the medium term? What are you thinking of doing?
In the short term, getting through my thesis. So I just qualified this last couple months here. So I need to put together a committee, a thesis committee, as well as really define what my work is going to be moving forward. Up to this point, I had been doing these large screens, and trying to figure out what my next steps were going to be. And it's getting to the point now that in the short term goal is figure out what I really want to do, and what my thesis is going to be about, and then formulate the committee around that, that I think is going to give me the best chances of success and make me the best scientist that I can be. And then in the long term, I really enjoy mentoring students. So my current PIs both know that. And so the fact that UT Southwestern doesn't necessarily have an undergrad campus, we do have things like the SURF program and other things that get undergrads here for research fellowships and such, but it's not like we have a large plethora of undergrads for you to interact with. But both of my mentors know that I'm really interested in that, and so they tend to give me rotation students to mentor for their first couple months in lab. And so I have a partial long-term goal right now to potentially look at starting my own little lab on an undergraduate campus so that I could get the opportunity to both teach and mentor students, as well as have potentially a more established lab and some more graduate students for some of the higher tier work.
Cool. And then I guess just to finish up, what has the last 2 years during the pandemic been like for you? So it's been, a trying time for everyone, but sort of an unusual time to be a virologist. So how has it sort of affected you personally and professionally, I guess?
Yeah, it's absolutely been interesting. I've had a lot of really unique experiences and life changes during COVID. When lockdown started, I was still a technician in the orchard lab. And I felt a little guilty not getting able to complete quite as much as I'd hoped to do before I had to leave for grad school. I'd already been accepted and my timeline was there, and we were just kind of trying to get through everything we could. And unfortunately, I didn't get through quite as much as I would have liked. But then I got to start grad school in the middle of a pandemic, which was also really weird. So everything was kind of online. A lot of teaching was online. And unfortunately, learning things in the lab and experiments from 6 feet away has its own challenges, but we persevered. We got through it, and we're doing a lot better now. Texas is doing a lot better, and on campus, we're really great around campus about managing our risks. And being a virologist was both rewarding and detrimental, I think. Though I don't necessarily study SARS-CoV-2, I got a lot of questions from friends and family because they know I work with viruses. And so I both got a little bit perturbed because I wanted to make sure I was up to date with the literature and what was right. I didn't want to be leading them astray. But also I got to do what I love, which was teach. And if you're teaching the ones you love and you love what you teach, then it was just a really, really great time. And I found that super rewarding. So it was not all bad. Yeah, thank you so much again for having me. And I hope to meet a bunch of people that potentially listen to this the day of.
This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, and other podcast providers, or at lmtv.podbean.com.